April, you're working on what at least I would consider, and I think the majority of the industry would also consider to be sort of the next gen, right, of RNA therapeutics, especially as it comes to gene editing tools, relying on linear mRNA, relying on novel RNA modalities like circular RNA. So I'm really curious to have you address sort of the same question that Greg started out with here with his linear mRNA. How would you characterize the health and maturity of the supply chain for RNA-based gene editing products as it relates to your drug substance, right, and your drug product?
Yeah, for linear mRNA, thankfully, for the in vivo editing application, we can really piggyback on where the vaccine industry has moved recently. So that's, like Greg was saying, there's lots of options there. For these gene editing applications, it tends to be more orphan drugs, smaller patient populations, so the scale is not as big of an issue, I think, for the vaccine space, or as it is for the vaccine space. So I think that supply chain is fairly mature, with a lot of folks on platform processes now. Lead time can still be a problem, and trying to get into the clinic, and that's a big barrier, especially when you're trying to sync up getting your plasmid and your mRNA together, because the timeline starts getting quite lengthy. And in the gene editing space, like with the gene therapy applications for mRNA, we're often pushed to make, say, GMP master cell banks even early on, which is not something the vaccine industry has to consider when you've got things changing a bunch. They're not pushed as hard to make those GMP masters. But if you've got a consistently made product, we do have to deal with that. The IP landscape is a minefield.
I've heard that. I've heard that.
Every inch of it has some sort of problem related to IP, from the caps, the untranslated regions, the nucleotide modifications that you use to stabilize it, the start initiation codons. It's trying to figure out how to navigate that, to have freedom to operate or pay for it, and the cost of that is tough. Circular RNA, I don't think it's really being made at scale or GMP yet, or at least not that I've seen. We do make it internally for iPSC freezing, so that's kind of where people are starting. So there's still something to go there. And for guides, it really depends on length of the guide. And this is a very different process, but the length matters.
Okay. So there's a lot, right? There's a lot we're navigating here. And especially, I think, as a small company, right, that you're challenged as well to both not only source these supplies, but also to navigate that legal landscape, which is something that not often is easy to do when you already have resources that are constrained, right, in terms of a small company and expertise. And I would also sort of extend this question that I'd asked Greg as well to you, in that, from an availability standpoint to capability, right? Are you finding that your partners can help you out, right, with stuff like the legal aspects of sourcing your materials, or how are you navigating, right, weighing the quality of the vendor partners that you're working with today in terms of getting all of the materials that you need?
Yeah, well, they do vary a lot. There aren't regulations or a gap in this space at the moment. That won't be the case forever. But lacking those sort of consistent regulations and pharmaceutical standards, everybody does things a little bit different. And like I say, when it comes to the IP landscape, some are willing to commoditize or democratize. That's a common term these days, like whether people want to democratize their IP and give you easy and relatively cheap access to it, or whether people really want to hold on to it. And you find both in this space. So you have some vendors that are like, well, we have kind of an FTO option that involves the vaccinia capping enzyme, but it's not scalable beyond 100 liters. Versus if you try to get in with caps, it's the whole mess. and sometimes they're like, I don't even want to deal with that. Like, you bring the cap to us, you work out the IP situation, like we're not going to do that.
You work out the IP situation.
They vary a lot of their appetite for really helping out, and there's some areas their legal department just said don't get involved. So the caps are a big problem, I think. And also because of the cost that they go in. And then I think for the editing industry as a whole as well, there's been a lot of advances in what works well for vaccines.
Okay.
But the optimal RNA molecule or mRNA molecule with regard to the nucleotide modifications to stabilize the PCRs and things that will work best for editing, that's still being worked out.
Completely
So I can't go to a supplier and say, well, what nucleotide modification should I use, like 5-methoxy-UTP or is N1-methyl-pseudo-UTP okay? Like they say, well, we have those options, but we don't know for your
We're not sure yet.
We're not sure yet. So there's a lot of things we have to check and trial and make sure that, you know, obviously we would want to go with something like N1-methyl-pseudo-U because it's cheaper.
Yes.
There's not as much fee around it.
Yeah.
So we don't know if we can. Like we have to check all of this stuff. Kind of to know. And then when I think there's still a gap when it comes to the sterile boundary. So with vaccines, with mRNA that's going in as a drug product, it's pretty clear cut. You have your traditional drug substance, which would be low bioburden. You then put that into an LNP, and that's your drug product, and that needs to be sterile. That's the normal way of doing things. But when you're looking at ex vivo applications of gene editing tools, you know, you can't sterilize cells. You can't filter. You can't, you know, generate. There's nothing you can do with the cell. So the sterile boundary actually moves a whole step. So we have that issue with the cell therapy products where if we're using mRNA or guides, and we'll have to use both actually to make the edit, that stuff has to be sterile. And currently manufacturers are operating as API suppliers, so they're low bioburden. So we have to deal with when it shows up in our shop, we've got to figure out how to make it sterile and make it okay to add to the cells. So that's an extra step for us. Have to do that. That's
That's a really good point. I think that ties really nicely to what Greg was saying, is it really, because we are still such an early industry, even I would say still on the vaccine side of things, right? We're still trying to broaden the availability and ability of mRNA to function as a vaccine across different indications. And if we're looking at the therapeutic realm, since we're still so early in understanding how our products work, right? And what makes them up and what's important and what's not. I think, you know, yes, right? What works the best. I think, you know, Greg mentioned a lot of the onus does come down to the manufacturer to figure out, right? Or to you guys, the biotechs, to figure out, okay, well, if we do try this material, what's it going to do, right? what are we trying to figure? What's it going to, what impact is it going to have? So, you know, Greg, I'd like to turn the discussion to you as well, because I know you do also work outside of the linear mRNA space. I'm curious if you have anything to add to what April has shared on the therapeutic side.
Yeah, for sure. I mean, obviously, most of what I currently work on is linear mRNA for vaccines, but, you know, for sure, a lot of exposure to these next generation mRNAs and next generation delivery as well. I do want to comment. So, you know, April made some really good points on IP. It is definitely worth noting a lot of the foundational IP around mod-UTP and mRNA in general and some of the early lipids, surprisingly, are almost at the end of their shelf life, right? So keeping that in mind of when you're going to commercialize your product. But of course, a lot of the next generation work and some things that are specific to each product, maybe, you know, a custom lipid for delivery, are certainly, you know, the differentiating factors. But, you know, in terms of, you know, maybe from a supply chain, raw material perspective, as we think of either self-amplified mRNA or circular mRNA, I mean, there are, beyond some of the obvious advantages in terms of dosing, in terms of performance, which is maybe out of scope here, from a manufacturing perspective, I mean, some things get simpler, right? Circular.
We can hope.
If I didn't have to worry about my capping and tailing, it definitely would make my life easier. There are. So I think a lot of what we laid for groundwork for supply chain will be very beneficial there. And on the flip side, there are also some nuance, maybe a little bit different purification, or the delivery, the LNP, the best LNP for linear might not be the best for some of these other modalities. So you do have some new materials that will come into the fold.
Yes.
You know, but again, they're for the most part, these are the same classes of materials. So you can at least bank on some of the infrastructure that has been built for all of the raw materials and starting materials and excipients that we've kind of built